All right, we're going into the next chapter, which is biotechnology. This is actually pretty interesting. It's not a process that involves polymerases and step 1 and step 2, etc. So this is kind of how biology is applied. And every single day, you guys deal with stuff that is based around some of this basic biotechnology that's come about since really the 70s, since the 1970s is when it started to take off. So I'm going to talk about, I'm kind of picking and choosing some of the main aspects of biotechnology and then some of the really important applications in your lives as far as biotechnology goes. So we'll kind of just, this is mostly kind of more surface level information. It's not going to get as in depth as, you know, like base pairing rules and, like I said, all of the extreme detail that we went into before. So kind of a step back, broader view of biology. All right. First, we have to start by talking about recombinant DNA. So recombinant DNA is kind of like what it sounds. Basically, you have a DNA molecule that's made up of 2 different sources. So like 2 different organisms' DNA is put together. This can be really simple, and I'll talk about sort of how it started with bacteria. But it was the first recombinant DNA molecule was created in the 1970s. So before that, they didn't really have the ability to combine different organisms' DNA. Since then, it's taken off and really made a lot of strides since the 1970s, since that first recombinant DNA molecule. So why do we do this? Well, there's a number of different reasons that I'll talk about at the beginning of this PowerPoint, but it allows us to isolate and manipulate DNA. So you can figure out what a specific gene does if you take it out of one organism and put it in another and see what effect it has, that kind of idea. That's a very simplified version of it, but it totally revolutionized biotechnology. So it changed everything. Yeah, exactly. Or from an animal into a plant, that's done. Yeah. And I'll talk about some examples of that. They started out pretty small. So they started with viruses and bacteria. You can learn a lot about what a specific gene does if you take it out of one organism and put it in another one that didn't have the ability to create that protein or whatever it was in the first place. And that brings us to cloning. So this cloning, I'm going to talk about reproductive cloning, which is what most people think about, like making an entire cloned organism. This cloning is just cloning DNA. So you're basically growing DNA or cloning it using bacteria. So to do this, they use something called restriction endonucleases. These are just enzymes that will cut DNA at very specific points. They're naturally occurring. So these endonucleases are normally found in bacteria. Bacteria get sick, which we don't normally think of. So viruses can invade bacteria. And they've, for, God, probably millions of years, been fighting each other, bacteria and viruses. So bacteria have these restriction endonucleases that will essentially chop up the virus DNA. So viruses have DNA, if you did not know that. And the bacterial endonucleases chop up the DNA, which basically makes the virus inoperable. If you chop up the genetic material of something, it can't invade the cells and reproduce like viruses do in living things. So it's a way for the bacteria to fight viruses. Biotechnology experts have been able to kind of utilize these endonucleases and cut DNA at the specific points where they want it cut. Obviously, there's a lot that goes into figuring out how to do that, but they can take a chunk of DNA out of a bacterial a plasmid. So bacteria have circular DNA. I think I've said that before. It's called a plasmid. It's just their DNA. It's just not that double helix. It's a simple circle. So they can take a chunk of that out and then insert a new chunk of DNA into it. And I'll show you in a very simple form what that looks like on the next slide. And they call this a vector. So then that bacteria can reproduce that DNA over and over again. Yes?
Can viruses do the same thing to bacteria?
Not that I know of. No. Good question. I don't think viruses have endonucleases and they can't fight bacteria. I don't know how they fight back against endonucleases. That's a good question. So yeah, the bacteria will then replicate this recombinant DNA with the new little segment of DNA that's been inserted in there. Bacteria just grow by splitting into two. So no sexual reproduction, just asexual reproduction where they just split in two over and over again. So you can get a lot of bacterial cells with this new combination of genes. So this new DNA, essentially. That's how you get the recombinant DNA. So this is kind of what it looks like. You can ignore the ampicillin resistance gene. We're not really talking about that here, but you can see what this restriction endonuclease is going to do. It's just a protein, comes in and cuts out part of that yellow piece. So we'll imagine the yellow piece is something they want to get rid of. Insert in foreign DNA. Obviously that involves a whole process as well, but in a very simple sense, the restriction endonuclease chops out a little segment of this. This is the plasmid, so bacterial circular DNA. Take part of it out, put the new DNA that they want in there. So we have the DNA inserted with the green gene in there. And now that bacteria will just reproduce over and over again. So that DNA will be in all of its offspring. Yes? Yeah, it doesn't always work perfectly. So there's definitely rejection issues. And I imagine that's probably at the very beginning when they tried to do this, a huge problem. So they had to figure out how to get it to actually stay in there. Other enzymes are involved to actually glue all that together. Yeah. So this makes it look really easy. You just take a chunk out and put a chunk in, but that's the level we're gonna stick with. And gel electrophoresis. So there's a couple of different processes I'll talk about. I won't go into too much detail on either of them, but these are two. Gel electrophoresis is one of the two really important methods for being able to separate out different segments of DNA. So in order to separate out the DNA, first off, DNA is negatively charged, so that's important. You can put the DNA in this gel. That's why it's a gel electrophoresis. So you basically extract DNA. We're not going to talk about how that's done, but just imagine extracting DNA. Load it into these wells at the top of the gel, and then this substance here is just a clear gel. You hook it up to electricity, basically. Because the DNA is negatively charged, this end is positively charged. So once you start running electricity through there, that DNA will start moving down and separate out. So you get this separation of these different fragments of DNA based on their size. So you can't tell a whole lot about it, but you can at least tell what the big sections are, what the small sections are, just based on where they land. The bigger sections are going to move a lot slower just because they're heavier. And then the smaller segments will move faster. So if you run it for, say, 20 minutes, an hour, whatever, the smaller fragments will move a lot further down towards the positive pole. You can then actually cut the DNA out of there if you know kind of about the length you're looking for. The length of the fragment you're looking for, you can cut that out and actually amplify it using that DNA cloning that I mentioned before. Gel electrophoresis is used for a ton of stuff. So you'll probably maybe at some point hear about this or see it, but that's the basics of it. It's just electrical charge, making these different length fragments separate out from each other. So you can separate out segments of DNA. So yeah, then to reproductive cloning. Like I said, I'm not really talking about reproductive cloning. I guess it is a biotechnology. But because we were talking about the micro or molecular cloning, just cloning the DNA, I thought, let's talk about reproductive cloning because usually people ask about it. So reproductive cloning is, molecular cloning is where you're just having bacteria reproduce, which like I said, they just split in two. It's really simple. Reproductive cloning involves the cloning of a multicellular, usually sexually reproducing organism, like a sheep. So you guys remember Dolly the sheep? That was a really long time ago for you probably, okay? That was like the early 2000s. So she was the first animal that was actually a clone. And I think it was in England they did this. So what they did in order to create a cloned sheep is they took an egg cell, took the nucleus out. So they needed the egg cell because it has a lot of yummy nutrients and stuff, the developing embryo, eventual developing embryo. But they don't want that nucleus. They want the nucleus from the somatic cells, which are just like the rest of the body cells of a sheep. So they took mammary gland cells. Those have nuclei, right? So they have a nucleus. So the DNA is in there. So basically the clone came from this this sheep whose somatic cells were taken. So take a somatic cell and then combine that with the egg cell just to get all of those nutrients basically into the somatic cell. Shock it with some electricity. That will make them fuse together. And then that'll eventually develop into a zygote and then into an embryo, and it'll be implanted in a surrogate. So Dolly came out of that. The first, like I said, multicellular cloned animal. There was lots of, and there still is a lot of ethical debate about if this is good or not good. Not IVF, no. So IVF is using, you would basically use the egg cell and with the nucleus, but you're not combining it with another cell. Yeah, good question. So she was exactly the same as this sheep. So there was no mixing of DNA. It was all the DNA from the somatic cells of this sheep are in her. She doesn't have any like father DNA. Yeah. So IVF still has that combination of mother and father. So yeah, that's reproductive cloning in a nutshell. She didn't live very long. She only lived like 6 years because they took the somatic cells from an older sheep. So they think that might have affected it. I think other organisms have been cloned since then, but that's the kind of first seminal one. Anyway, that's reproductive cloning. Back to technology here, other technologies. Reverse transcription. This PowerPoint is kind of just a mishmash of a bunch of different biotechnologies. Reverse transcription is the next one I want to briefly talk about, where it is exactly as it sounds. So normally transcription, you're moving from DNA and making RNA. Reverse transcription is where you're taking an RNA molecule and making DNA from that. So the point of that is basically to kind of isolate the part of the DNA that actually goes to code for the protein. So if you remember, a lot of the DNA doesn't code for anything. It's just kind of in the way DNA, and the RNA has to be kind of cut down and processed after the transcription process to get to that final coding sequence for the protein. So by going backwards, you get rid of all of that noise within the DNA. So you just have the sequence that codes for that protein, so you can learn more about the protein that way, or learn more about the whole process that way. So the DNA that's created is called complementary DNA or cDNA. So if you ever see cDNA, you know it's from this reverse transcription process. So going backwards in the central dogma, basically. Okay, and then another really important technology you may have heard of, if not, you may eventually hear of, is the polymerase chain reaction, or PCR. So PCR, if you've had a COVID test done and it's been a slightly longer one, so not like an hour turnaround, they used PCR to actually amplify the viral DNA to see if you had COVID. So PCR is used all the time in a lot of different scenarios. It wasn't developed until the 90s, but what it does is basically produce a ton of DNA. So it will copy the DNA really, really quickly rather than having to wait for the bacteria to reproduce on its own. This will just really speed up the process. So it's a really powerful tool in biotechnology these days. It uses a DNA polymerase enzyme called Taq polymerase. So Taq polymerase, this is the enzyme that puts down the new DNA nucleotides basically. And it's from, interestingly, that's a bacteria that's found in hot springs. So like the cool looking hot springs that you see out in Yellowstone. That's where these bacteria that have this enzyme are found. They use this because they actually do this at really high temperatures in a PCR machine. So they need something that can handle really high temperatures, and Taq polymerase can handle those really high temperatures. There's 3 basic steps to PCR. First, so if we're trying to just copy the DNA and make a ton of copies of that one strand of DNA, we start with the original double-stranded DNA at the top. And then that has to be separated or denatured, right? So we know that's the first process of DNA replication. If you think back, way back to learning about DNA replication, you got to unzip the DNA. So denaturation happens at a really high temperature, 95 degrees Celsius. And that's just below boiling. So 100 degrees Celsius is boiling. So this is really hot. That's why Taq polymerase is used, since it can handle those high hot springs type temperatures. And then the primers have to be attached at a very cold temperature, -50. So it drops it down in this machine. It's at 95, and then it drops it down to -50. The RNA primers are laid down, which starts the whole, eventual DNA strand. And then the third part is the DNA synthesis. So that Taq polymerase comes in and then adds all those new nucleotides to grow that strand. And now you'll have 2 copies of DNA, where before you just had the 1. And that happens at 72 degrees Celsius. And that's about 116 Fahrenheit. So still a really hot temperature. And then you get just a ton of copies of DNA in a very short time period. You don't have to wait, like I said, for the bacteria to reproduce on their own timeline. All right, the next technology, DNA fingerprinting. Have you guys heard of DNA fingerprinting? Yeah, a few. So this is kind of what it sounds like, using part of a person's genome to identify them. So like a fingerprint is just part of your identification. You don't have to sequence an entire genome, so all the DNA of somebody, to know if, say, they were the perpetrator of a crime, or it's used in paternity tests as well to know if they're the father of a child. So investigators basically use it to identify an individual based on a small amount of DNA. So maybe only a small amount was left at a crime scene, or paternity, they can get more DNA, but it's just a quick, easy way to identify a person. And it's based on these unique parts of our DNA. So we have a lot of our DNA is pretty similar to each other, but there's these short tandem repeats, STRs, that vary among individuals. So those are what they're honing in on. That's kind of the fingerprint of a person. So they'll hone in on that, sequence those short tandem repeats, and try to match them up with either the child in the case of a paternity test, or a suspect in the case of a crime. This uses gel electrophoresis, that process we talked about, to match up the DNA. So it doesn't even look at the specific sequence. So it's not like it's looking at TACG, that sequence. It's just looking at the general structure of the DNA as far as the size of the fragments. And I'll show you what that looks like on the next slide. But this is where gel electrophoresis comes back into play. So we have a crime scene on the left. and we run that DNA that we found at the crime scene through a gel electrophoresis, and we get these bands. So these are just different fragments of DNA, different sizes, right? So this is a really short fragment. That's going to be a really long fragment because it moves a lot slower. And that'll match up with, ideally, hopefully, if you have a suspect, one of the suspects whose DNA you've also taken. And if they match up, this isn't perfect. There are errors, but if it matches up compared to two other suspects, then you can have a pretty decent idea that that might be the person who committed the crime, if we're talking about a crime scene. So DNA fingerprinting is used a lot in criminal investigations. And this is a way that now they can go back and they're kind of looking at old cases where maybe just an eyewitness was used, but there's DNA. They didn't have this ability before to kind of do the DNA fingerprinting, and people will get released based on the fact that their DNA didn't match with the crime scene DNA. So DNA fingerprinting is really important if you're interested in anything like forensic. It's a huge technology that's really come a long way. All right. Now, into something I mentioned briefly, but I mentioned that we can now edit genes in a living cell, so in a living person, which is mind-blowing. So this hasn't been possible until maybe 5 years ago, not even 5 years probably. So this is really, really recent new biotechnology that is going to essentially change the world. So change medicine, change the world, change how you're treated medically from a genetic standpoint. So yeah, until recently, we haven't been able to alter DNA in a living cell. And now it's possible. There's still a lot of work to be done, but the system that's used is called CRISPR-Cas9. So CRISPR is probably what you'll hear most often, and you might come across this just in the news. It's getting more and more attention. It stands for something really long, which is why they shortened it to CRISPR. Clustered, regularly interspaced, short palindromic repeats. So it's kind of like those STRs in the fingerprinting. So just repeated sections of DNA. These are stretches of DNA that are found in bacteria. So back to bacteria. And they're, again, used by bacteria to fight viruses. So a lot of this biotechnology goes back to this battle between bacteria and viruses, and we have utilized that in order to help ourselves, essentially. So the CRISPR, along with the protein, which is called Cas9, cuts up viral DNA, and oftentimes will insert new DNA in its place. So the Cas9 protein cuts up and destroys DNA of invading organisms. And I have a video about how this works a little bit better that will hopefully explain this a little bit better. Scientists have now figured out how to basically guide that Cas9 protein to, say, a gene that's not functioning correctly. So say sickle cell anemia. If that one base has been replaced and mutated to cause sickle cell anemia, you guide the Cas9 protein to that segment. It can chop up what it needs to, the incorrect sequence of DNA basically, and put in new DNA. And now from then on, that person may not have sickle cell anymore. I don't think it's been used at that level yet, but that's kind of the direction that we're going. It's really amazing. So DNA can be, here's just some, basic bullet points about how this, some simple bullet points about how this works. DNA can be repaired or replaced. The mutated form of the gene can basically be replaced by the normal gene sequence. So especially if it's some, like sickle cell, debilitating genetic disease, it can be fixed just by kind of recoding the DNA. That's huge. Yes?
How is it mutated? Like what is able to mutate it so that you can put it back to where it needs to go?
How is the DNA mutated?
Yeah.
So there's a sequence, it'll kind of show in this video, but there's a sequence that will kind of guide So you have the DNA and it'll bring in an RNA molecule that matches up with it. So it'll match up with the mutated version of the RNA or of the DNA. Yeah, and then cut that out. Yeah, so we can target it to activate, repress gene expression or identify. It doesn't even have to be used to actually cut up a gene. You can just use it to find a specific gene. So it helps to locate specific genes within the genome, which can be difficult. So the two ladies who actually developed this, there was a lot of people working on it at once, but they were the first to kind of figure out how to use this as a gene editing tool. They just won the Nobel Prize in 2020. So this is really, really new stuff.
I have a question.
Sure.
Okay, so is the CRISP, like, is the Cas9 shorter for whatever that
Oh
stands
no,
for?
so CRISPR is CRISPR's the DNA like the DNA sequence, and then Cas9 is a protein that'll cut the DNA where it needs to be cut.
Oh, okay, thank you.
Hopefully this video will help explain it in a little bit better than I did. So we're gonna watch this video real quick. Maybe. Okay. Oh, come on, sound. There you go. Okay. So this is Jennifer Doudna. I think that's her first name. Dr. Doudna. She works at Berkeley. So she's one of the two women who developed this and yeah, won the Nobel Prize last year for it.
About science and about biology for me is that there's always more to be. The thing that's amazing about science and about biology for me is that there's always more to be discovered. And every time we feel like we've figured something out, it seems to open up several more questions. Scientists have appreciated for a long time that once we understood the DNA sequence in cells, if we had a tool that would allow easy manipulation of that sequence, that would be a very powerful kind of technology. CRISPR is a technology for changing the sequence of DNA in cells in a precise fashion to correct mutations that might otherwise cause disease. It's going to enable a lot of science to be done that was impossible to do in the past. So the way that CRISPR technology works is by the action of a protein called Cas9 that functions like a molecular scalpel for DNA. The CRISPR-Cas9 system has an amazing ability to recognize a particular DNA sequence in a cell that may be malfunctioning and then disable it by cutting the DNA. We call this gene editing, and we can use it to disable or repair a mutated part of the gene, which may be causing disease. For Cas9 to find the malfunctioning DNA, we attach it to an RNA sequence that matches the DNA sequence we want to edit. Then we put this RNA-Cas9 combination into the cell. It finds the mutant DNA and uses a chemical reaction to cut the DNA strand right at the spot where it's malfunctioning. After that, we can sometimes insert the correct version of the gene for the cell to work properly again. It's a very exciting technology that's going to do a lot of good in human society and for human health. It stretches from human therapeutics to agricultural applications to thinking about how do we make better biofuels. These would be incredible outcomes, I think, of using this kind of technology in the future.
All right. I don't know. Good question. The whole process. I know they use, yeah, they use PCR some to amplify the DNA, but I don't know, yeah, what it looks like how they get the DNA into the cells or the Cas9 and CRISPR into the cells. Yeah, I don't know enough about it to go into that detail, but that's the basically how that CRISPR-Cas9 works. So if you see CRISPR, now you kind of know what that is. You may or may not. I actually I think I heard multiple podcasts about CRISPR before I put together this lecture. So it's definitely out there, like in the public kind of science realm. All right. PowerPoint, yeah. So we'll get more into this on Wednesday. I just want to introduce the idea, of GMOs. Everyone's heard of a GMO, right? I think at this point in your lives. They get a lot of praise and they also get a lot of disdain from people. So GMOs are just genetically modified organisms. We'll talk a lot more about them on Wednesday, but this is a huge biotechnology that has been around, well, for a while now, but gained a lot of interest, both positive and negative, in more recent years. So GMOs are any organism that's been genetically altered by some process other than conventional breeding. So for thousands of years, we've been breeding plants to make bigger fruits, or we've been breeding animals to help in the fields. So that's not considered GMOs, but we have been altering the genetics of organisms for a long time. GMOs are more pointed, I guess. Yeah, that doesn't involve breeding. It involves some kind of molecular alteration of the genome. So DNA can be just modified, changed somehow. It can be deleted, can be added to, or you can inactivate genes. So GMOs kind of include a bunch of different aspects. And I think I want to go into a little bit of detail on GMOs because I think people sort of have a general idea of what they are, but I think, as biology students, it's good for you guys to really know what a GMO is. Transgenic organisms is often what they're called if they have DNA from multiple organisms put together. So some plants have bacterial DNA in them in order to make them pest resistant. So that would be a GMO, and that would be a transgenic organism. So something with another type of DNA other than its own DNA in it is called a transgenic organism. And this is possible because, like I said, the genetic code is universal. So when you look at that chart of the codons where you find the 3 letters and figure out what amino acid it codes for, it's universal. Like I said, across at least all eukaryotes, even some prokaryotes. So you can figure out the same code is going to code for the same amino acid, which will eventually make the same protein. So it's alterable because it's universal. It can help scientists understand genes' functions by creating these GMOs. It also they have really important applications, especially, and I know we don't think about agriculture that much, but agriculturally, GMOs have made a huge impact on the agricultural landscape. We eat every day, so agriculture is important to everybody. Oh yeah, so I'll end on this slide. So one kind of early-ish example of how organisms were modified genetically is by creating these knockout organisms where, in order to understand what a gene did, scientists basically silenced a gene or knocked it out and replaced it with another gene, or just knocked it out to see what effect it had. So it helps scientists understand, like I said, what the gene does. If you take it out and then see what the result is, then you can hopefully have a better understanding of what that gene does. So it's been used to study a lot of different stuff. Really important stuff: cancer, obesity, Parkinson's disease, aging, substance abuse. And then it also, I found this particularly funny and amusing. So this gene that creates myostatin helps to regulate muscle growth in mice as well as humans. So we have the same gene. So if it's knocked out, if it's normal, the mouse looks normal. If it's knocked out, the muscles basically just grow out of control. So you get this really ripped mouse that you see here. So these really big chunky muscles. So it really helps you understand what that myostatin gene does when you get rid of it. So something simple like that. There's a bunch of examples of these knockout organisms that are used to study different genes. But I thought that was one of the more amusing images. A chunky little ripped mouse.
Is it the same mouse?
I don't think it's the same mouse because the whole gene was knocked out, but same species, yeah. Okay, and we'll stop there and go into more GMO stuff on Wednesday.
What time is our lab on the third? I mean, our final.
Final?
Yeah.
110. Yeah, I think it's 110.
This is